Okay, so now that we've established that association, we have our organism growing in pure culture, now we need to put it back into the same plant. And this is step 3 of Koch's postulates, where we re-inoculate the same plant, species, or variety with the organism that we now have in pure culture. The goal is to reproduce the symptoms of that disease on our newly inoculated plant. Now that we have a pure culture of the organism from the diseased apple, we need to complete step 3 of Koch's postulates, where we determine if this organism is capable of reproducing the symptoms of the disease in a healthy apple.
For this step, we'll need a scalpel, alcohol matches, and an alcohol lamp, an apple of the same variety that we isolated from, paper towels, the pure culture that we've isolated, parafilm, and a digital camera and notebook. You'll need to document the appearance of your pure culture. To do this, you can either describe it or take a photo that you can insert in the lab notebook. We are going to inoculate the apple by making several small wounds and then putting plugs of the hyphae into those wounds. By wounding the apple, we are creating favorable locations for infection to take place. Begin by surface sterilizing the apple with alcohol. Then sterilize the scalpel. Now we'll make several small wounds. Make sure you mark which wound is going to be the control and which ones are going to be inoculated with the pathogen. The first thing we'll do is flame sterilize the scalpel again. Now in one of the wounds, we are going to put clean agar that does not have any organism growing in it. This will serve as our control to ensure that any effects are caused by the organism and not by any of the procedures that we are performing. So take a small piece of agar away from the colony. And then carefully place it in the wound. Then reflame the scalpel. And this time, we're going to take plugs from the colony and place them into the other wounds. Flame the scalpel again for the next plug. We will now put parafilm over the inoculation sites to ensure a favorable environment for infection. We will now incubate the apple and observe daily for symptom development.